Hi,
Navjot,
everybody. Yeah, hi, everybody. It's my
Good
pleasure to
afternoon.
see the faces on the screen. They are old friends, not by age that they are old, but by our relationship. One is, of course, Albert. We know probably each other for almost last 14, 16 years. We met long back. Terry, I usually keep close contact with him, and we are by profession we keep in maintaining the relation. Of course, Sangeeta is like my sister. Ranjit is my good colleague. Then Bipin, we are colleague. So without spending much time, what I will do, because it's a diverse, we have got different from different countries. We have got participants. I will just tell you about it AATM for our friends, because we have grown in last 22 years. We started in 1999. Now we are almost 23 years old. Not me, the organization. So after that, now why we are growing? Now we have got 23 member countries. Actually, we work like a United Nations. Our boundary is from Turkey to Indonesia and Central Asia, like Uzbekistan to Philippines. This is the area we operate. 3 WHO region. These are WIPRO, AMRO, and CRO. And thanks to all our friends like Sangeeta, Ranjit, Sadhana, so we have got all dedicated members, and we are it's going forward. Just to give a short, this webinar being organized by one of the working group, that is got working group called Plasma Protein Therapy. And they are doing excellent job, very good. But because there are experts like Albert, expert like Terry, Ranjit, and in this 23 member countries, this is very badly needed, and we are in contact with everybody, including, I can tell you a few names. I can see Rekha is here, like it's in Nepal, because recently I had a WHO assignment in Philippines. They need this. I mean, very, very sad to say, almost 90% to 95% plasmas are thrown away because there's no facility, even for contract vaccinations. So having saying that, I feel this webinar will be success. I'm here, I'm joining you. Number two, we want your cooperation to channelize plasma in this part of the world so that we can do something. Why to throw away so many plasma, precious human resource? So I leave it to you. This is the mandate of our working group, that is working group on PPT. Without taking much time, I request Sangeeta as well as Ranjit to take it forward. Thank you, everybody, and thanks to all the delegates as well as speakers to give their consent to join this webinar. Thank you very much.
thank you, Navjot, for introducing AATM to this forum. I will request Sangeeta to say a few words before I really start formally the webinar.
Thank you so much. And again, a very good evening, good morning, good afternoon, as per the time zones that we all are here today with. And I extend a warm welcome to all of you for this webinar. And my extreme thanks to Dr. Navjot Chaudhary for putting up such motivating words and encouraging words for our team. And these motivations keep us going. And it is because of his strong leadership, we all know that the AATM has reached to these heights. And today we are sitting with all people from all around the globe because of his extreme efforts that were done to form these working groups, especially the plasma protein therapy group, which otherwise was unheard of and is a very specialized group which is working towards the usage and the PDMPs of plasma that we are seeing. And today we are going to discuss that. And I'm extremely glad that we all are here today for this webinar on the relevance and importance of plasma-derived medicinal products in peace and war. It's a very relevant topic to be discussed in the times that we are seeing today, which are around us. And we have a very impressive line of experts from all around the globe, which you all can see right now, and who are going to enlighten us on the subject. So once again, a welcome to all of you and my best wishes for the success of this webinar. Thank you so much. Over to you, Ranjit. Thanks.
Thank you, Sangeeta. First of all, I would like to thank all the speakers, Albert, Theri, Padma, Bipin, and unfortunately, Antesha, I don't see her there. All of them were very gracious. They consented to come on this seminar. And they bring a different kind of experiences within the plasma domain itself. To start with Albert, starting from TGA head regulatory perspective, to the patient perspective, to the technical, to the PPTA, to the IPFA. I think the list can go on and on. His contribution to plasma industry is simply unbelievable, and he has done so much good work on the technical side, on the regulatory side, on the patient side, and of course, PPTA and IPFA. It all brings so much experience. And I think if one Googles out his name on the plasma, probably he will take 50% of the presentations and the publication on plasma side. So such a rich experience Albert has on its side, and I'm sure we will be greatly benefited by his presence. Same thing for Theri, he has a very, very impressive career. I see Antesha has also joined. I see Theri has also contributed so substantially in the field of plasma from France, then moved on to Taiwan, and he's doing such a good work, so many presentations. He has remained up with WHO as a consultant, and he has been a good friend like Albert, and we meet quite often on various plasma forums. So his contribution is very, very immense into the plasma space, and he brings a lot of technical expertise, how to help in the contract fractionation, and very recently has started some work in Taiwan as well. And I hope, Theri, with that, we are able to do something in this part of the world. Also, Padma is there with us. Padma basically comes from India, but settled in the USA for a long period of time. And she has done good work on the source plasma collection. She runs few centers in Florida. And I think one can debate on the source and recovered plasma, but plasma is needed by one way or the other to help the patients. And I think she also brings an experience how to collect the source plasma, what are the challenges, opportunities, etc., etc. Bipin is there from Nepal. He has been working into transfusion medicine space. And also, he has been talking to me on the plasma, setting up a plasma fractionation program, either contract fractionation or setting up a plant. But I think Nepal is also pretty much into that space of plasma thing. And I think we need to work on that side also. Antesha has joined from Ukraine, one of the most politically challenged territory right now. And I would like to especially thank her joining at this moment. Though I reached out to the business manager of biopharma company and he connected me to somebody else. And then, it came out and she brings experience from the clinical side, which is equally relevant. You start the product, you start the plasma, you make the product, you regulate it, and eventually use for the patient. So we can see the galaxy of people with us bringing experience from product to the regulation, to the manufacturing, and to the, at the end, clinical use of the products. So I'm extremely glad and thankful to all the speakers who consented for it. Just to bring the things in perspective, what Navjot has said a few minutes back, that AATM is a forum where we are trying to help not only the member countries, but anyone who would like to work into the plasma space, whether to help in the increasing the quality, enhance the quality of plasma, collection of plasma, storage of plasma, or contract fractionation, or whatever best we can do. And I think this is the best speakers available in the world who can help us how to have these member countries and countries around us to make, of course, the self-sufficiency is a bit of a difficult term to handle it. It depends on the product, it depends on the geography, etc., etc. But how to help the patient, I think that's the bottom line we are talking about. And how to minimize the wastage of plasma, how to harmonize the regulations, etc., etc. I think that is the mandate. We would love to work with it, and cooperation, help will be greatly appreciated from each one of you And this will certainly help patient, whether they are in A geography or B geography. Patients are patient and they need the medicines to survive. And I think that is how we all are focused to work on it. And this is also the mandate of working group of AATM, which is PPT, plasma protein therapy. So thank you very much, one and all. And to start the thing, I would like to request Thierry, who is basically from France, but these days is in Taiwan and is working on various blood plasma products, on platelets, etc. And I would request Thierry to enlighten us with his talk, and I'm sure it will be helpful to all of us. Over to you, Thierry.
Okay, thank you, Ranjit. Let me arrange my screen. Oops.
Oh, your slide is pretty much visible.
Yeah, you can see. Yeah, okay.
Yes,
but,
yes.
Just want to remove this. Okay, now I can. So thank you very much for all the introduction from Dr. Nachub, Dr. Sangita, Dr. Ranjit. I was very happy also to see Albert on the screen. I have not seen him for some time now. So thank you again for the invitation to talk at this webinar. And the topic Ranjit asked me to address is access to safe plasma products with some Asian perspective. So let me see. Yeah. So the first question I ask myself is, why do we address this topic for Asia today? And I think the reason maybe are mentioned here, that many PDMPs are essential and unique therapeutic products. There are global shortages of affordable PDMPs in the world, in particular in low and middle income countries, which are most affected by these shortages and putting patients in Asian country at risk, in particular in these Asian countries which do not have access to a plasma fractionation program or a plasma fractionation infrastructure. And these shortages are, as we know, amplified during global or local crises like pandemics, war, natural disaster. And these impact the collection of plasma and the capacity also to carry out plasma fractionation and deliver the product. So this puts patients, in particular those with bleeding disorder and immunodeficiency, at risk, especially in low and middle income countries. And these patients do not have sufficient access to affordable treatment. And the World Federation of Hemophilia repeat that the situation does not appear to have changed for over 30 years. So there may be some issues which are faced in order to improve the access to plasma products in Asia. There is an insufficient volume of domestic plasma in Asian countries, and plasma meeting quality requirements for fractionation. And there may be different reasons why this is the case. And also in many Asian countries, we are facing a fragmented blood collection system, which does not help to organize a plasma fractionation program. There are logistic and contractual difficulties to organize a contract plasma fractionation program in some Asian countries. Although we should note that several of the countries in the region have been successful to implement a contract fractionation program. There is in several countries no access to the traditional plasma fractionation technology, and this is in part linked to the fact that it is quite a complex industry. I like to say it's among the most complex industry in the biotech field. And countries may be faced by lack of local expertise and skilled manpower in order to address the manufacture of plasma derivatives. And then some countries are facing a lack of regulatory and blood policy system to support safe and effective access to plasma and plasma products. So having said that, let's now have a look at some characteristic of the plasma fractionation technology and look at why it's difficult to organize and to manage a domestic plasma fractionation facility. If we look at the current plasma fractionation technology, the one which was developed many decades ago and which little by little evolved over the last years, you will see that the technologies which are used at large scale still involve cryoprecipitation to recover cryoprecipitate and make factor rate in particular, combined with chromatographic procedure to isolate the most fragile proteins like coagulation factors and protease inhibitor, and the traditional ethanol fractionation process still used in most centers to make albumin and under decreasing use for the production of immunoglobulins. But you can see that the standard batch size or pool size for this industrial plasma fractionation process may involve 2,000 or 6,000 liters of plasma for each pool, which is quite a substantial volume, difficult to achieve for some low and middle income countries in the region. What are the features of the plasma fractionation technology which creates problem for countries to implement it? As we said, the large plasma batch size for each pool, between 2,000 and 6,000 liters, roughly. You have under the same roof the plasma fractionation facility, several lines of fractionation to prepare multiple products, which implies particular working procedure to avoid cross or downstream contaminations. You have technologies which combine purification step and viral reduction treatments for inactivation and or removal, and this is integrated within the fractionation process to purify the plasma proteins, and thereby the engineering of the plasma fractionation industry and plasma fractionation technology is quite complex in order to cover the purification of different type of proteins by different type of technologies, the production of utilities and their distribution. So all these require very specialized companies capable to handle this complex engineering. And understandably, considering what has been happening in the past with viral transmission, this industry became probably among one of the most, if not the most regulated in the world to guarantee the quality of the starting plasma for fractionation and the safety of the PDMPs, which is to be considered to be the same at global level. So the requirement when we want to consider a domestic plasma fractionation facility has to consider the fact that it is regarded that the targeted yearly volume of plasma required to consider building a fractionation facility should be up to and more than 300,000 liters. Needs to find a knowledgeable and reliable supplier of technology, capable also to train the future local manpower, find the skilled engineering company for the reason we mentioned before. There is a need for heavy capital investment, which is sometimes very much underestimated, at least at the start of a project. Need to identify skilled and stable manpower. And of course, there should be government support and a regulatory system in place. So this volume of plasma is hard to reach in many of our countries in Asia, in particular the smaller countries. Finding a supplier of technology is possible. It can be identified in Asia or Asia Pacific, and local expertise may exist and can be expanded. And engineering company with a biotech industry experience could be suitable to tackle the particularities of the plasma fractionation industry and fractionation technology. So when domestic fractionation is not achievable, then there is a possibility to engage into contract or toll fractionation, which has been an option followed by some Asian countries. And how is it done? You have the plasma which is collected in the country. This plasma is typically centralized in one place where it can be shipped to a fractionator which is located abroad. Then products needed in the countries collecting the plasma will come back and can be made available to hospital or to patients for home treatment. We should keep in mind that there are several kinds of contract or toll fractionation scenarios which are possible and which can be discussed with the plasma fractionator. I will not detail those now, but maybe we can come back on this later if needed. So the requirement among others to initiate a plasma fractionation contract involve the fact that, first of all, the plasma quality must comply with the specification of the fractionator and their own regulatory requirement where the plasma is collected, which implies also that the blood establishment should be subjected to auditing by the plasma fractionator. There should be a licensing of the blood establishment by the local national regulatory authorities, which is collecting the plasma, or where is collected the plasma for fractionation. There should be a licensing of the contract fractionator and the fractionated products by the local national regulatory authorities where the plasma is collected. And of course, there should be in some countries a need to obtain authorization to export the plasma for contract fractionation and for the import of the PDMPs. And obviously, the product portfolio made by the plasma fractionator should meet optimally the requirements of the supplier, the control of the supplier of the plasma, such as low dosage formulation for factor VIII or immunoglobulin, preparation of fibrinogen, which is very often needed in some low and middle income countries, preparation of prothrombin complex, etc., etc. The parties involved in a plasma fractionation agreement involve the need for a good communication, good understanding between national regulatory authority of the countries supplying the plasma and the countries where the plasma fractionator is located, as well as discussion, communication between the plasma supplier and the plasma fractionator in order to ensure the reciprocal auditing between the two. So the advantages of such contract or toll fractionation system is a good means to use domestic plasma and avoid the wastage, as was mentioned by Dr. Chaudhuri. It provides some guarantee of stable supply of PDMP. I would not say self-sufficiency, but I would say at least having some important product or essential product made available to patients locally. It helps to improve the sustainability of blood establishment through optimal use of blood plasma, and at the same time, the system in place to improve the quality of the plasma for fractionation through auditing and licensing. At the same time, helps to improve the quality and safety of red blood cell and platelet for transfusion or plasma for transfusion. I think a contract and a toll fractionation agreement is also a way, or it can be seen as a learning phase on how to improve the quality criteria of plasma for fractionation. So, as we said, benefiting the quality and safety of all blood components in the blood establishment collecting the plasma for fractionation, and then spreading to all other blood establishment in the country. And it can be seen also as an interesting learning phase where the future operators of the local facility can be trained by the plasma fractionators on quality assurance aspect, quality control of plasma products, manufacture of factor VIII immunoglobulin, good production of the utilities. So it can be very helpful when there is a potential technology transfer for domestic fractionation. Then now let's move to another scenario, which is local improvement and small-scale processing to improve patients' treatment using locally available plasma. And here maybe I will describe things which are of particular interest for Asian countries with relatively small volume of plasma and relatively small population of patients. This, what I'm going to say is taken largely from a recent WHO guidance document, which, as I remember, was published in March 2021. You have here the link where you can have a look at this document, and I extracted 3 chapters from this document. One which is on how to implement strategy to collect plasma for fractionation, either the use of recovered plasma from whole blood or the use of so-called concurrent plasma, which is a byproduct of platelet pheresis, and when needed, the introduction of apheresis to collect plasma by this dedicated technology. Then you have a chapter on the production requirement of plasma for fractionation, which provides a number of important details to make sure the plasma can meet requirements of plasma fractionator. And then you have one chapter, which is quite new at the level of WHO, which is the recognition that in some country and when plasma products are not made available or are too expensive or are not in sufficient supply to meet the needs of the patients, then a stepwise approach is something which can be considered to prime the system to gradually improve the situation instead of being completely in a dead end and not being able to do anything. So this WHO guidance has been relayed by the Global Blood Safety Working Party of ISBT. We organized a workshop in September 2021, and 2 additional publications have been made to report the main finding from this workshop, and you can see the link here if you want to have more details. But basically, what was mentioned and discussed during this workshop is the fact that when it comes to process local plasma, we have to take into consideration numbers. And the first numbers which we discuss is the fact that currently it is considered that in order to attract the interest of a plasma fractionator to perform contract fractionation for another country, there is a need to have at least, I put here 20,000, but actually during the workshop, we talk about 50,000 liters, at least 50,000 liters per year on a consistent basis in order to consider the fact of engaging into a contract fractionation agreement. And here the upper level of 250,000 liters is mentioned because you are reaching a level of a volume of plasma which may justify the fact of moving into the construction of a domestic facility. But then the dead end is, what do we do when a country has less than 50,000 liters of plasma per year? What to do not to waste the plasma? And this is where the idea came that it would make sense to provide safer therapies to patients who otherwise are not treated or are treated with unsafe crude plasma fraction like non-virally inactivated fresh frozen plasma or cryoprecipitate. So this is a way which was seen as being a good means to provide stepwise improvement where it is needed. And it is something which can be achievable and which can also complement industrial fractionation. So the two can live together for some time, and it's a way to prime a system in order to step by step go towards industrial fractionation and preparation of industrial concentrate for treatment of bleeding disorder or immunodeficiency. So among the techniques which were discussed during the workshop was the fact that it can make sense, and when it is cost effective and technically achievable to perform pathogen reduction treatment of plasma, with the techniques which are licensed and mentioned here, as it is a way to be able to prepare cryopoor plasma as a source of pathogen-reduced material for treatment of several deficiency in coagulation factors or treatment of TTP patients thanks to the presence of ADAM13 in the cryopoor plasma. Or then with this pathogen-reduced plasma, you can make cryoprecipitate as a source of safer factor VIII, fibrinogen, virulent factor, or factor 13 to treat these deficiencies in coagulation factor, either acquired or congenital. Then this workshop and the WHO document refer to the possibility to perform a mini pool solvent detergent treatment of cryoprecipitate. So this is something which was developed in Egypt by Dr. Magdi El-Kiabi, and which basically what you do is you pool 30-35 units of low volume cryoprecipitate, completely depleted of cryopoor plasma, and you process by a treatment of viral inactivation by solvent detergent, followed by oil extraction to remove most of the solvent and detergent, followed by activated charcoal filtration to remove impurities and residual solvent detergent. Then you have a sterile filtration, and then you have a product which contains factor VIII, coatable fibrinogen, virulent factor, and factor 13, which can be labeled on the units of cryo which are prepared from this pool in order to provide regular prophylaxis and better treatment of inherited or acquired bleeding disorder. I think it may be a solution to consider in some Asian countries, and something which we can keep in mind, that this type of process is scalable. So it is a step towards smaller scale plasma fractionation and larger scale plasma fractionation. And the same is true for a technique to prepare pathogen inactivated, virus inactivated immunoglobulins, thanks to a treatment based on caprylic acid purification and virus inactivation, and with a process which includes a concentration step, which allows to get a product with 60 milligram per ml of protein or immunoglobulins, which can provide a source of safer immunoglobulin. I would like to mention that this type of process may not be in a PAC system, but has been introduced or used in Pakistan to prepare SARS-CoV-2 immunoglobulin. Again, a situation something to consider to prime local IgG production is something which is scalable, and it is a step towards contract fractionation or domestic facility. And when the scale is expanding, then additional virus reduction steps are readily can be implemented relatively readily. So then I come almost to the end of my presentation, mentioning the launch of the International Coalition for Safe Plasma Protein. So again, coming back from the WHO guidance document followed by the GPS workshop, this was one of the conclusions, to try to create an international organization which could help a stepwise development towards plasma fractionation. This is an organization under the leadership of ISBT, but with important international organizations like the International Federation of Blood Donor Organization, the IPOP, or International Patient Organization for Primary Immunodeficiency, the World Federation of Hemophilia, and as associated member, the IPFA and the PPTF. And actually, there is one project which is ongoing now in Dakar, Senegal, the National Center of Blood Transfusion, under the leadership of Saliou Diop, for the introduction of a mini pool SD treated cryoprecipitate for treatment of fibrinogen deficiency, factor 13, and patient with factor 8 or virulent factor deficiency. So finally, the take-home message of my presentation for this webinar will be that there is a continuous increasing need for plasma products globally, and most particularly in Asia. The shortage in plasma products affects especially patients with bleeding disorder and primary immunodeficiency in Asia, and this is true for low and middle income countries and to some extent high income countries. we have seen that various technological options exist to enhance the supply of safe plasma protein products and PDMP from domestic plasma in Asian countries. And we have seen that stepwise technological approach from mini pool to large pool is something which is now new, but which may be meaningful to improve patient treatment in some Asian countries with insufficient supply of in PDMPs and with a relatively small volume of plasma, which prohibits them to do contract fractionation or domestic fractionation. And with this, I would like to thank you for your attention.
Thank you, Thierry, for very, very detailed, very elegant presentation. And I think you made a very valid point, Thierry, here. And I remember meeting you in 2002 in Lille. And coming from there, you know, when I started my work way back in 2001 after returning from the US, I was not able to collect 10,000 liter plasma in 2004 in India. From length and breadth of this country of 1 billion people, with a great degree of using all my skill set, I was able to convince Green Cross Korea to agree to do the contract fractionation of Indian plasma. And initial volume agreed was just 10,000 liter. And unfortunately, I could only send 8,500 liter plasma. And so journey began from a very small bit of a plasma. And now India collects about 600 to 700 or maybe 800,000 liters of plasma, close to 1 million. So I think all baby steps, what you have suggested, are really very important. And of course, there was no proper regulation in India. There was no plasma policy, hemovigilance program, NIP, etc., etc. So I think your presentation does bring all those elements, what is needed to improve the quality of plasma, contract fractionation, regulation, and not to underestimate the capital investment and the HR part of it. So thank you very much, Thierry. We will keep the question for the end. And I would request all the audience over here, kindly send your question in the chat box, and we will be happy to direct your question. Are you will have opportunity to ask question directly towards the end of this webinar. So Thierry, once again, thank you very much for this detailed presentation. May I request Albert now to kindly, take us through with his wonderful slides and the great work what he has done over here. Albert, to you.
Well, thank you very much, Ranjit. Good afternoon to all of you. I say good afternoon, but for me, it's actually 9:10 in the evening. I'm speaking to you from Melbourne in Australia. I would just like to share my screen. Okay, share, and I just need to do this. Okay, are you able to see the full screen now?
Yes, absolutely, Albert. Please go ahead.
Okay, well, first of all, thank you very much, Ranjit, and my dear friend, Dr. Chaudhry. I have very fond memories of the first time I visited you. It must be over 20 years ago now, when you were working together. And it strikes me that many of the issues which we are talking about this evening, we talked about in those first meetings. And thank you very much for your kind introduction, which is totally, in my opinion, undeserved. But anyway, look, I gave some thought to thinking, what could I say to add some value to this meeting, which perhaps it would not be said otherwise. And I want to share some personal reflections on issues which are affecting the field as I see it now. Some of these issues perhaps are not very directly impacting on your country, but I think that as time evolves, you will find that they will. And in fact, this evening, we've already heard some of these issues mentioned, as I shall show. So I'm going to take each of the three main products. Of course, there are many more, but I'm going to take each of the three main products from plasma fractionation: albumin, immunoglobulin, and factor VIII, and discuss a specific issue related to each of them, which is actually applicable throughout the field. If I think about albumin, which is a product I've been involved in now for a large number of years, since I joined the PPTA. Before that, I wasn't really very interested in albumin. But since joining the PPTA, of course, I'm no longer in the PPTA now, I gained an interest in albumin. Albumin, as you know, was the very first real mass-produced plasma derivative. It was the result of the excellent work by Professor Cohn's group in Harvard University, and very quickly became the driver for procuring plasma globally, and stayed that way for a large number of years. Albumin initially was used to save lives of people who had suffered severe blood loss, in battlefield injury, in burns, in trauma of various kinds. As the years have gone by and intensive care management has improved, the use of albumin in these circumstances has actually become less easy to justify, because with good modern technologies in relation to intensive care and emergency medicine, we can actually manage volume loss quite well in most patients, not all, with other solutions, including crystalloids, which are much less expensive. However, in the past 10 years or so, a number of so-called pharmacological benefits of albumin have been explored in relation to the albumin molecule's complex structure and its ability to exert various physiological actions in the body, including suppressing oxidant stress and things like that. And one of these indications has been very vigorously pursued in our countries, in the Western economies, and this is the use of albumin in severe sepsis. And what I'm showing you here is the basic summary of 3 very large randomized clinical trials. The SAFE study in Australia, which was carried out about 18 years ago. The EARS study, which was primarily driven by France, and the ALBIOS study with primary investigators based in Italy. And what you can see here is that when you compare survival between the groups who received albumin and the groups who did not receive albumin, always we get an improvement in survival with patients with sepsis in those who received albumin, but this is never reaching statistical significance in these large individual randomized clinical trials. And there is one study, the ALBIOS study in Italy, which showed that in the subgroup of patients with septic shock, you got a certain statistical difference in survival, but not very large. However, if you look now at this most recently published meta-analysis, which takes those studies which I have just shown you together with other studies and combine them in a meta-analysis comparing benefits with albumin and benefits with crystalloid, you can see that effectively there is no statistically significant difference. And basically, patients with sepsis do equally well overall in randomized clinical trials combined in a meta-analysis when they are given crystalloid, which is something to think about in relation to sepsis. If we look at another indication, which is the use of albumin in long-term administration in patients with cirrhosis, this is the famous ANSWER study from Italy in Bologna. And you can see in this study that survival in patients, long-term survival in patients given albumin as well as standard medical treatment was significantly enhanced compared to those not given albumin. This study was published in The Lancet. It is a randomized clinical trial, and it caused much interest some years ago. If, however, again, we look at a meta-analysis of patients from many studies in terms of them being given albumin in cirrhosis and ascites, you can see that again, we have no difference in significant clinical benefit in terms of this other indication spurred on, fueled by the possibility of exploiting albumin's tertiary structure. And last of all, in terms of albumin, we go to the established indication. A lot of albumin is used in this context, especially in the United States in cardiac surgery. You can see, looking at this retrospective, I emphasize a retrospective cohort study in the United States, you can see a very significant benefit using albumin compared to crystalloid. But again, when we look at a randomized clinical trial, as opposed to a retrospective cohort, this study from Finland, published a little earlier this year, shows no significant benefit. Why have I chosen albumin to make this point? The point I'm seeking to make is that plasma, as I think has already been mentioned by Thierry and others, is a very precious and very scarce resource. And we need to make sure that plasma is used to the best possible benefit for patients in terms of the products which are derived from it. And this means that we must always question indications, and we must also always practice evidence-based medicine. This is something I firmly believe in, and I have given you this example of albumin in the context of my first message to you, which is that clinically in transfusion, we must continue to pursue the best practice evidence and continuously do clinical trials. I now go to the important issue which has been mentioned by Ranjit, which is the issue of self-sufficiency. In our economies in the West, the plasma supply, as everybody knows, has been driven for at least the last 15 years by the apparently never-ending increase in the demand of immunoglobulin. And much plasma is collected for immunoglobulin production, and at the moment, globally, in Europe, in the United States, everywhere where plasma is collected by the big industrial fractionators, we hear the slogan, we need more plasma for immunoglobulin. Let us look at this interesting example in terms of a small country in Europe. This is the Czech Republic. It is a relatively small country. It's a very nice country. I have visited it many times. The capital city, Prague, is one of the most lovely cities in Europe. It is a reasonably prosperous country. The GDP per capita is nearly $28,000 US. And believe it or not, my friends, this little country is the biggest producer per capita of plasma for fractionation in the world. On a per capita basis, the Czech Republic produces more plasma than anywhere else. The United States, other European countries such as Germany and Austria, they all lag behind the Czech Republic. Now, if you look at the Czech Republic's plasma collection, and this is data for 2021, which is the latest that we have, you will see that, as I said, there is this enormous volume of plasma which is collected compared to the population. 860,000 liters of plasma is collected. And if one had to take out of this plasma in the immunoglobulin using a yield of, I think Thierry would agree, of between 4 and 4.5 grams per liter, we can expect that the Czech Republic would have at its disposal 375 grams per thousand population of immunoglobulin, which in terms of all that we know about the indications for immunoglobulin is more than enough for the Czech Republic's patients who require this product, much more than enough, probably 2/3 to 1/2 more than enough will be generated. Is this happening? No, it is not. You will see from this slide that the very latest figures from the Czech Republic show us that rather than 375 grams per thousand of inhabitant, as we would expect from its plasma collection, the Czech Republic is consuming 70 grams per thousand. Now, I would say that 375 grams per thousand is too much, but 60 grams or 70 grams per thousand population is too little. The Czech patients are underserved. And the reason for this is very simple. There is no link in the Czech Republic and other countries which collect plasma, and I think that this is a very important point, between the actual collection of plasma, whether it is from the commercial paid donor sector or the voluntary recovered concurrent plasma sector. There is no link in the Czech Republic between this vast collection of plasma and the provision of product from this plasma to the Czech patients. The Czech plasma is all of it sold to the fractionators who use it to make product, which they then offer on the open market to various countries, including the Czech Republic. And since there is no link and no public health infrastructure in the Czech Republic to ensure that the Czech plasma derives benefit for the Czech patients, like there is, for example, in Italy, this is not happening. And the reason why I put this point to you is to remember that self-sufficiency is not just a case of collecting more plasma. There has to be public health policy driven by the government to ensure that a community's plasma actually goes to benefit that community and not end up as a tradable, saleable commodity on the open market, with all that entails for countries which do not necessarily have the funds to buy expensive immunoglobulin. I think that that is the second important point I wanted to illustrate with immunoglobulin. I now come to a point which has already been covered to some extent by both Dr. Chaudhry, both Thierry, and also mentioned by Ranjit. And this is the case of hemophilia. I'm going to talk about hemophilia A and what I call the tragedy, the continued tragedy of waste. This is the global prevalence of hemophilia A in terms of numbers of patients, estimated in the most recent publication from the clinician Alfonso Iorio, who is Italian, but who works in McMaster University in Canada, one of the most able minds in the field of hemophilia and evidence-based medicine in the world today, a close friend of mine, and I admire him greatly. And he has used a sophisticated statistical methodology, which I personally don't understand, but I trust his estimates, to estimate that actually the global prevalence of hemophilia A is more than we previously thought, and it is over 1,125,000 patients are suffering in the world from hemophilia A. Now let me show you this slide, which shows you, and this is a slide from the Market Research Bureau, which we all depend on for data on the plasma industry, very reliable, very wide data, and generously put in the public domain. And this shows you the global collection of plasma for fractionation over the past 20 years. And you can see that it is continuously going up. You can see that there is a dip in the COVID years. It went down in 2020 and in 2021. My understanding is that this year, thanks mostly to the United States, because the vast majority of global plasma for fractionation in the world is collected in the United States, it is going up again. But the data we have here shows us this dip. But irrespective of that, you can see in this slide that there is a certain volume of plasma collected, about 70 million liters. And you can see that the vast majority of this plasma increase in plasma collection in particular is from the apheresis source plasma, most of it, not all, but more than 80% of it from the commercial sector, whereas the recovered concurrent plasma mentioned by Thierry earlier seems to be always stable and stagnant at that relatively low volume. Now, I have done a very simple set of calculations in relation to this plasma for fractionation which is being collected, and I estimate that if with proper management, all this plasma, most of which is suitable for generating rich cryoprecipitate, is fractionated to factor 8. And again, I look to Thierry for comment on my estimation of yield, but I think that 180 IUs of factor 8 per liter is a reasonable figure to aim for. And if this was extracted, we would have nearly 12,000 IUs each patient for each patient in the world per year more than we have. And remember that many of those patients estimated by Alfonso Iorio are not actually diagnosed. So the amount of factor 8 able to be distributed from this hypothetical source of plasma cryoprecipitate is much, much more for the patients we know about. Is this happening? It is totally not happening. The vast majority of the cryoprecipitate generated from plasma from fractionation globally, unfortunately, is discarded because thanks to the enormous, wonderful advances in biotechnology in the rich countries no longer use plasma-derived factor 8 for hemophilia A. And rather than some kind of mechanism, and we have much thinking to do about how to do this, and it is possible, I firmly believe, to channel this cryoprecipitate. But in the meantime, tens of millions, hundreds of millions of international units of factor 8 for patients with hemophilia A are being thrown away. And this is a global scandal. So I have reached the end of my thoughts, which I wanted to share with you. So I just want to summarize my basic ideas. I think that in transfusion medicine in general and in plasma product usage in particular, we need to use evidence-based medicine because plasma for fractionation is a scarce resource and we need to make sure that we are using it wisely. And one of the ways is to make sure that optimal use and indications which are recognized are what it is used for. I have shown you that it is not enough to proclaim, let's collect more plasma and let's get self-sufficiency, because unless there is a mechanism in the public health and within governments to ensure that there is a link between plasma collection and provision to patients, this plasma will simply flow towards the commercial sector and will find the best price irrespective of where it came from. And we have many examples for this. And finally, I have shown you one example, but I think there are several more whereby fractions which do not find a use in the developed world are being discarded to the great detriment of patients worldwide. And with that, Ranjit, I thank you very much again for your introduction. And I will stop here and look forward to the discussion later on. Thank you very much.
Thank you, Albert, for very, very thought-provoking. And these slides were really very, very interesting from the point of view that how the industry and what are the challenges we are facing and what it could lead to potentially. And I guess your slides are very critical and important in the sense of the usages of albumin and factor 8 non-availability and immunoglobulin. And I think there has to be some logical sequencing needs to be done on the plasma collection and making it fractionation and available in the public health domain. And I totally agree that, Albert, when I was talking to you way back, a project was to come in India in public health space to make use of Indian plasma. Unfortunately, it never took off within the bureaucracy of the system. But I think you raised a very, very valid point that plasma collection is not good enough until and unless it is linked to the fractionation and eventually public health policies facilitate its trickle down to the last person in the queue. So thank you very much, Albert. And I'm sure you
I just wanted to make the point, Ranjit, that the slides obviously now belong to the society and they are freely available from my point of view to be distributed to anybody who is interested in them.
Sure. Thank you, Albert. Thank you very much, Albert. Sure. Thanks, Albert. So it was a great to, see your presentation after a long time. And thank you once again, Albert. I didn't know you are in Melbourne. Somewhere I thought you are in Europe. I'm sure it's...
Well, Ranjit, I can show you the reason why, but I don't want to waste time. However,
Sure.
I can tell you that 5 months ago, while I was in Italy, because that's where I live most of the time, I
Right.
became a grandfather.
Oh, congratulations.
And the young lady lives in Melbourne. So I came
All
to
right.
Melbourne to see my new girlfriend.
Sure, sure. All the reason for you to be in Melbourne, Albert. Thank you very much. Thanks. Now, I would like to invite Padma. She has been running 3 plasma collection centers in Florida in the US. And I visited one of her center in Jacksonville about 2, 3 years ago. And she will bring a perspective of source plasma collection and how it is being done, what are the challenges, how it can be done, regulation, etc. are one of the important part, at least in this part of the world. But I'm sure she will throw some light on source plasma collection. So Padma, over to you.
Thank you, and I really appreciate you to, including me in this atom that first time I got to know, and I didn't know this one exists like a 23 nations membership organization. And again, once again, thanking for the opportunity to speak about the plasma collection centers, which is the 95% collection from the world. And I'm a very small business plasma collection center, and Himaras is started my husband, Dr. Jairam Chigurupati, and by myself. And it is compared to all the big plasma collection centers in US, I'm slowly growing the Himaras LLC, and which is a FDA approved facility in 2015, and we follow FDA guidelines, collecting every drop of the plasma. Again, like other experienced professionals mentioned, it's a plasma is the there is no alternative. Human to human, we need to save is all the derived coming from the plasma. But after COVID, plasma collections are drastically dropped, like a 50% of the collections. Now, like it is bounced back 20% more collections in the US, and I think 95% of the collections that coming from the US, that is true fact. And the other countries have the ethics and barriers, not able to start a plasmapheresis centers. And also they have the, like you mentioned, the bureaucracy of each country is different. Like Malaysia, they want a kosher plasma. India, you can't pay donors to collect the plasma. So whole world depend on the US plasma collections. So how do we overcome? That's still in, it's an ongoing, and somebody has to represent the plasma and then open the doors, and it should be self-sufficient for their own country, and also manufacture the plasma derived products from the own country. So again, the Himaras LLC is a 14-year-old company, and compared to all the big players like Grifols, Acta Pharma, and BPL, I'm the small compared to them, but I still running this plasma collection center, and FDA license, and a woman-owned business, a plasma collection, which is very challenging to sustain in this business. For example, when I went to the PPTA last month, the cost per liter to collect the plasma, it from $50 went up to $300 because donor remuneration fees went up drastically. So it's a challenging to be in this plasma collection. Because I'm not in the fractionation, but I'm in the only collection, plasma collection. So there is a dollars and cents, and the collecting the plasma and the other speakers are focused on the IVIG, all that. But on the other end, the beginning of the manufacturing is depend on the collection of the plasma, which is very expensive. Like for example, India, they can't afford to that this kind of expensive plasma import to India. My point to say that is India should have their own plasma centers, and hopefully Indian government able to understand this. Compared to recovered plasma, source plasma is the best plasma to get the manufacturing, get the more IVIG, more grams per liter. Recovered plasma, you lose a lot of the during the fractionation, during the filtration, you lose in the especially Crohn's method. My point is like Asian countries and all, which should be have their own plasma collection centers. It's much more viable at this point of the time after COVID. There is a pre-COVID collection centers, and another point I want to make. In 2008, when we started the humerus plasma in the US, there's only 450 centers. Now they have almost 1,000, more than 1,000 centers plasma collection in every state of the United States. FDA see the importance of the plasma collection, and they are vigorously approving the new plasma collection centers. So because of that, I was able to expand one center to the 3 centers in state of Florida, and in the plans expanding to at least 11 to 12 centers in next couple of years. So that's the plasma collection side of the overview or perspective. I can now Europe also opening the plasma on their own plasma centers like US. They don't want to depend on the US plasma collection, and also Canada opened up 5 centers. This is the first time. Like India, they don't have the ability to do collect the plasma because it's not without paying. It's non-compensated plasma. Now the Canadian government approved to pay the donors collect the plasma from Canada. So the compared to last 10-15 years, the perspective of the plasma collection centers is changing, and hopefully we can get an approval in India to open a plasma center. That's my perspective of the humerus, and then how I visualize the plasma collection and then help the Indian population.
Thank you, Padma. You brought in a very important perspective from the point of view of the source plasma and what are the challenges. Unfortunately, not unfortunately, I would say the way West has evolved on the plasma collection and the regulation. Unfortunately, I mean, at the moment, India is yet to pick up, Asian countries are yet to pick up. And I've been on the regulatory side as well, and I realize it's not an easy job for them as well. The moment they open the gate for the source plasma collection, it may create havoc in the system if it is not properly regulated, if it is not properly monitored. And it needs a hell lot of effort to develop that regulatory system and monitoring system before really one can allow the game to start. Otherwise it can be really very, very chaotic. And we, as Albert had said, there are multiple stories available that, if it is not properly regulated and properly not maintained, the cost is a patient's life, right, at the end. So I think it's a good thought to look into the source plasma collection in this side of the world. And yeah, a large part of the plasma does come from the US. But of course, this is a valid point which all of us need to think about it and how to take care of it. I think that's the $1 million question which we will need to really take it forward. So thank you, Padma.
Exactly. So especially in India, the storage is the most dangerous part. It doesn't store properly. Whereas US has a stringent, as we speak, I am participating in one of our centers, FDA is auditing. So the way they do audits, the safety of the plasma, donor safety, the focus on donor safety and the quality of the plasma. If any SOP deviation, you get a 483. So we are always under constant fear and working under FDA. So the same kind of should be developed in any countries to open up the centers.
No, I think it's very, very important, because until unless we regulatory system and monitoring system is in place, we have seen enough of examples that it can create havoc. So thank you very much, Padma, and also especially to wake you up so early in the morning.
No, thank you. No. I'm
it's very
passionate
early for
about
you.
plasma, and then I can do anything in my, which I
Thank
can
you.
help or do anything. I'm
Thank you, Padma.
happy to do it.
Sure.
Happy
Thank
to
you to
do it.
wake you up very early in the morning in the US on the East Coast time. So thank you very much.
No problem.
Thank you.
And I want to thank you other speakers also. Maybe I may not able to say that, and I'm saying,
Sure.
in advance.
Sure. Thank you, Padma. I think with the presentation part now, Dr. Anthesia is there, and probably I would invite her. She comes from Ukraine, one of the most discussed, most covered territory at this moment, and she will bring a perspective. She is a pediatrician by training, and she will bring the perspective of PID. So, Dr. Anthesia, would you like to present?
Yes, hello to everyone.
Hi, good to see you.
Yes, I had some problems with connecting because we have problems with electricity, but I was able to connect via mobile internet.
Thank you so much.
I'm not a specialist in plasma collection or in fractionating, but I'm a clinician, and I will show you some slides about primary immunodeficiencies in Ukraine. So can you see my screen?
Yeah. Yes, we can see.
I'm pediatric and clinical immunologist, and we have a registry in our country, which is 42 million of people, population of Ukraine. At least it was before the 24th of February. Now we have a great migration, so we cannot calculate, but we hope that all our people will come back to Ukraine after the ending of the war. In our registry, there is 1,360 patients. More than 1,000 of them are alive. These include both mild immunodeficiencies and severe, but in total we have 825 alive patients with a severe PID. When calculating the incidence, we can say that it is 2.3 per 100,000 inhabitants. It is less than that it may be estimated according to the world incidence of about 10 live patients per 100 inhabitants, but we try to do a lot of work to diagnose new patients and to detect, to make new diagnoses, but it became not easy during the war. The antibody deficiencies are the most common PID, and this structure is similar to those which we can see from the world's registries, and especially from European Society Immunodeficiency Registry. So it is similar. Speaking about the need of the immunoglobulin treatment, I can say that about 80% of our patients require substitutional therapy with immunoglobulins. It is about 250 patients per year. These are not only antibody deficiencies, but also almost all patients with combined immunodeficiencies and some from other groups. 80% of them use intravenous immunoglobulin, and only 20% use subcutaneous immunoglobulin. But the way for the not only for the diagnosis, but also for availability, access to treatment, which is lifelong, was quite long and difficult, because immunoglobulin is expensive medicine. That's why me, together with patient organization, doctors, together with the patient representatives, spend a lot of time to have access to treatment. And one of the important points was the opening of the fractionator, national fractionator biopharma plasma in our country many years ago. This gave a significant advantage in this matter, because we use different products in our country for our patients, but those immunoglobulin products which we buy from abroad are several times more expensive than national product. And we have unequal geographical distribution of PID across the country. There are differences in diagnosis detection, but the first challenge which we had since the beginning of the war was the migration. Almost half of our patients went to other regions of Ukraine or abroad. About 25% went abroad from the country, and others were distributed within the country. Why it is challenging for us? Because we have a government supply for immunoglobulin treatment, and patient is connected with the hospital in his region, every patient, and thus we can plan the treatment when we know how many patients are in each region. We have 25 regions, so now it is quite difficult to plan. And the second challenge was that many hospitals were closed, or they were on the line of fire, so the medicines were closed in the hospitals, and the patients had no access to them. And now the last challenge is the problem with electricity. So, for example, I have no electricity now, and there are problems with the storage due to turning off of the refrigerators. This reflects the topic of this webinar concerning the peace and war, which is indicated in the title. This is the end of my slides, which I had to show to you. So I'm ready to answer the questions if I have not highlighted some things.
you bring a very different perspective to entire talk, not only from the clinical perspective, but the potentially the patients who are facing the challenge to get the medicine under the current political situation is very, very difficult, and you are operating. So I would like to really congratulate and wish all the very best that you are able to go back to where it is. And it's a beautiful country. I visited your country 3-4 times. I had great memories in walking in the streets and meeting all of you. So thank you very much for coming with all this difficult situation, making it possible. And you certainly bring a different view of the current situation, which probably, you are only facing it, and our all the best for everything from this side of the world and on the based out with everyone over here. Thank you very much once again.
Thank you for the invitation.
Sure. I think I have last speaker, Bipin from Nepal. And Bipin would like to, he has not, he does not have a presentation, but he would like to speak something about Nepal, what's happening on the plasma product side. So Bipin, over to you.
Thank you, sir. I'm Dr. Bipin Nepal. I'm currently working as a transfusion medicine expert at a international hospital. So we are in the first phase of developing plasma policy, and we are making the guideline. So we just need some of this presentation from all Dr. Thierry, Dr. Albert, Padma Shri, and everyone was very helpful. And we are just I'm very eager to know some of other points to be put in the guideline for contract vaccination agreement, like compensation financially or fractionated blood, how financially would be easy, and how government would discourage it, and other thing about our blood center is scattered in many parts of Nepal. So how the different quality measure should be taken and should be sent for the contract vaccination. So what would be easy for the government should control it, or any blood bank should control the plasma vaccination, like contract vaccination agreement. I want to know these things so that it will be easy for us to put it in the guideline. Thank you.
Thank you, Bipin. I agree that I have done it for India way back in early 2000, and it was quite a roller coaster ride, because the question, the challenges are quite locally confined, and we need to handle it with the local political situation. But I'm sure whether it is Thierry, Albert, or Padma, all of would be happy to help you out whichever way they can. And I would be happy to share even the presentations which I have with me. And anything which any one of us can help you out, I'm sure we'll extend all possible help and see how best we can see that Nepal also is able to make use of its plasma for its patient. And I think that is the most important part for all of us, to see that each patient in the Nepal is able to have the access to plasma product using the plasma coming from Nepal. So
Well,
thank you very much.
I'm sure.
Yeah,
that
please
Thierry
do.
I'm sure that Thierry can point to various WHO documents and others which he himself has contributed and formed over the past 20 years, which should be of great assistance to any country, particularly countries such as represented in this webinar. Thierry, why don't you just go ahead and point it?
Yes, thank you, Albert. I already put in the chat a link to the WHO requirement on collection of plasma control and regulation. So, yeah, production, quality control, and regulation of plasma for fractionation. So it's a pretty detailed and technical document which can give good information of all the elements of importance for the collection of plasma meeting requirements for fractionation, including issues with traceability, testing, and all these important aspects. And there is another WHO guidelines which is available, which is GMP in blood establishment, which since the collection of plasma should meet GMP guidelines, GMP requirements to be accepted as a raw material to make medicinal product, this GMP in blood establishment also is a good reference document. So if you go to your brother and you put World Health Organization GMP in blood establishment, you will find the document easily and can be downloaded. So I'm thinking about these two, Albert. There are probably others. There are what we call en memoir or things like that on on plasma fractionation, contract plasma fractionation, but these are not as detailed as the two guidelines I'm just mentioning now.
Yeah, no, I think Thierry, you made a very valid point, and I have shared my work, what I have done for India with Bipin, Rekha, and Manita. That in Asia, how in India when I started my work, what was the step number 1, step number 2, step number 3, which is more locally in the sense of relatability. So I'm sure with your inputs coming from Albert's suggestion, I think Nepal should be able to do it, but if any help is needed, as an AATM forum, we'll be more than happy to help them out, whichever way we can. And if needed, we can reach out to Thierry or Albert and anybody else who probably will be the relevant candidate at that point in time.
Yes, I mean, to some extent, Ranjit, the creation of ICSPP is also intended to provide some kind of reference or independent opinion for countries which are aiming at developing a plasma fractionation program.
Sure.
So this also ICSPP could be contacted.
Sure.
Yeah.
I think that the first thing which has to be done before anything else is to get conviction at the level of government that the procurement and generation of a domestic plasma supply is a national priority. Plasma is a strategic resource. At the moment, the world is in a situation of huge energy crisis because for many years of complacency depending on cheap gas from Russia, which today is being condemned for invading the Ukraine, but which has been an authoritarian dictatorial regime for many more years than has been the case in relation to the war on the Ukraine. And the world should not have allowed itself, Europe at least, where I spend most of my time, we're in a desperate situation because we allowed ourselves to not become self-sufficient in energy supplies, in Italy, for example, and be dependent on cheap Russian gas. And the analogy to me in relation to plasma fractionation is very obvious. If we continue this dependence on a single source of supply from the United States, we are totally at the mercy of, number one, the free market, whereby prices are cranked up according to what companies think the market can bear. And products move from one country to another depending on what price they can get. Number two, and we have an example historically which is very dramatic, the question of possible infectious disease in a donor population, which happened in the United Kingdom in the late 90s and kept plasma for fractionation out of that country for more than 20 years. So the first thing to be done is to convince governments that this is a necessary measure in relation to acquiring self-sufficiency in a very important, crucial resource, and not just to hand it over to the multinational private sector, because handing it over to the multinational private sector is not going to solve the problem of patients in India, patients in Egypt, patients in all these countries where there is a lot of plasma, but there is the risk that this plasma just flows out into the commercial world.
I think, Albert, you made a very valid point, Albert, here, that it is so important to have the political will to resolve this issue, and government must take as a top priority. And I agree, I just want to quote an example just from the context in India, that in late 80s, early 80s, what happened about the HIV with the factor VIII, So there were few fractionators in India, they were making factor VIII, but because of the HIV story, all the fractionation program was closed and there was no factor VIII available, no plasma available, and the patients were dying because of HIV infection, patients were dying because of non-availability of factor VIII, and so on, and other co-infections. Unfortunately, there was nothing happening, but one fine day, the chief secretary's nephew was diagnosed with hemophilia in All India Institute of Medical Sciences. And unfortunately, there was no import permit also at that point in time. For 4 years, 5 years, the patient suffered for the disease because of non-availability, because of the infection and co-infection and so on and so forth. But one fine day, 4 o'clock, this patient, young boy, was diagnosed with hemophilia A. And the very next day, the import permit was granted, which was not granted for the last 5 years. So this is, whether we want it or not, these are the political realities which we deal with day in and day out. And you are absolutely right that, there has to be a political will to make sure that it is a top priority and we should not be, taking it lightly. So thank you, Albert, for bringing it to the attention. Yeah, now the forum is open for the question and I see Ibrahim's hand is raised. So Ibrahim, please go ahead.
Hello, sir. I want to ask you and the other speakers about lyophilized plasma. Lyophilized plasma can be an opportunity in a war or in pandemics. What can you think about this? There are commercial preparats from Germany, from France, and from South Africa. And there are very good results in the literature about the lyophilized plasmas. It may be an alternative in war or pandemics.
Thierry, would you like to take this question? Albert, please go ahead.
I'm not sure I entirely understood. Is Ibrahim asking about convalescent plasma?
No, lyophilized plasma.
Lyophilized?
Yes.
Ah, okay. Well, I don't exactly see the relationship to pandemics, but it's actually quite interesting. For an old person like me, it's interesting the way that products which became redundant early in my career have come back. One very interesting example for me is fibrinogen, because when I started my career in transfusion, fibrinogen was on its way out because it was very good at transmitting viruses. This was before the development of viral activation. And because fibrinogen was, and still is, extracted from cone fraction 1, it transmitted a lot of viruses. But now it's come back. And the same with lyophilized plasma. It's interesting to me that there is a great interest in this product. Much of the interest and much of the funding in the United States is coming from the US military, which actually supplies enormous amounts of research money for various types of transfusion-related projects. I don't know what I can say about lyophilized plasma in terms of its accessibility in the normal transfusion context. Well, lyophilized plasma has to be reconstituted. Of course, frozen plasma has to be thawed, so it cuts both ways. And there are some technologies now involving spray lyophilization, which result in preparations which can be dissolved much more quickly. But I don't know that I have much more to say beyond that. What do you reckon, Thierry?
Yes, but basically I agree with you, Albert. Indeed, the interest for lyophilized plasma is mostly coming from the militaries, the US or other countries. And I know France is also preparing like pathogen-reduced freeze-dry plasma. And I think I agree also with your point on fibrinogen, which has been a forgotten plasma derivatives, which now is recognized as being of big interest in some part of the world, yes, and for acquired bleeding disorder. Yeah, I think I will not add anything more to what you said, Albert. Yeah. If I can make a comment, Ranjit, if there is no question. Yeah, I would like to comment to the slide which was presented by Albert. which is a bit shocking, where you mentioned that the factor rate is discarded, right? So factor rate collected from high quality plasma and being discarded. I think it comes back to the basic issues of the supply and the affordability of factor rate in low and middle income countries. I think the factor rate is not produced. It's the cryoprecipitate which is not processed into factor 8, because the processing cost of the cryoprecipitate into double valorly inactivated and freeze-dry factor 8 will come to a point where the industry will not make any profit by trying to distribute this product in countries in needs of this type of plasma-derived factor 8. So this is the big issues, and I know that there are donations from the World Federation of Hemophilia, but these are still very limited amount compared to the needs we are seeing in low and middle income countries. So this is again the issue of the cost, maybe the production cost of factor 8 from cryoprecipitate and not including the cost of plasma would be something nowadays between 15 and 20 cents of US dollar, which is already, and so at the production center, then you need to ship it away, you need to have a license in a country in need of factor 8. I think the industry is not willing to do that. They prefer to use their plasma and make profit on the sales of immunoglobulins, and they are not humanitarian companies. That's the issue, and that's the whole issue of the supply and availability of factor 8, affordable factor 8. This is why in my slide, I made shortages in affordable factor 8.
I agree.
In low and middle income countries.
However, there are pathways not very widely exploited and not very widely known. The best example in my mind is the country where I live mostly, in Italy, where the contract manufacture of Italian plasma by the Italian regione, by the regions, certain regions have directed the contract manufacturer to continue producing a surplus to their needs of factor 8 concentrate, which they then donate for humanitarian use. And this is a program which is administered by the regions and by the National Blood Center in Rome. In the other country where I live, where I'm at the moment, I am scandalized by the fact that we have contract fractionation in this country of 800 tons of plasma, which generates very high quality bulk cryoprecipitate, which is discarded because there is no political will. And this is the issue here. There is no political will. We have explored this, the stakeholders in Australia. There's no political will in government to generate a humanitarian aid program for countries in the region. Australia gives many billions of dollars in humanitarian aid to countries in our region, but there's no political will to utilize this possible source of humanitarian aid because there is an apprehension that there will be the perception that there's a commercialization of the plasma of Australian donors. In this country, it is only voluntary donors who donate plasma for fractionation. So what I mean to say is there are many pathways. Each country, each landscape has to be explored, and wherever possible, coalitions built in order to generate these pathways. I don't believe that it's impossible. Yes, of course, the companies are not going to generate lyophilized double viral inactivated factor 8 concentrate and give it for free. They'd never do that. But if there is a contract fractionation situation, there is the possibility to generate this, to generate more factor 8 from the plasma than is being discarded. And I think that it's one area which I would like very much to see more vigorous attention paid to in the specific countries, such as I've mentioned in Australia.
Thank you, Albert. I think you raised a very, very important point. And just to add what you said, way back in 2003 and 4, I visited the WPBTS, which is the Western Province Blood Center in South Africa. and they were making a wonderful factor 8, at a low economy scale. Unfortunately, that plant is shut down now. And I got trained on that plant, and it was making such a wonderful factor 8, but unfortunately, because of the political will and those related issues, it never took off. And eventually the plant is shut down. So I think it's very, very important for us to look on the larger landscape and how can we make use of all cryoprecipitate which is generated and get into more details of it and see how best we can help the patients. So I think it's very, very important. I think we have short the time with so much of interest from audience, and still there's any question, I'm sure we'll be able to answer it. But if not, I would like to thank everyone over here, audience and speakers, especially, Albert is there. I woke him up till late. And also Padma, I requested her to be up little early. So my sincere apology and thank both. And I see Navjot is raising his hand. I'll give him chance. So thank you very much, all of you. Yes, Navjot.
Yeah, Ranjit, my sincere request to all our friends, including the participant, not to end this seminar at this point only. Actually, we have to carry it forward. And as I talked to Albert as well as Terry, this is an acute syndrome, acute problem in this part of this world. Lots of plasma is thrown away. We are discussing, like Manita is here, she's from Nepal, then I have seen the name of Rekha also. They need badly this plasma fractionation or plasma sourcing. Problem is that process is slow. Ultimately, we are losing plasma every day. We know that in Western countries, it's plasma, they need plasma, but we are throwing it away. There is a question of, quality of plasma. That, of course, we are there to improve the plasma as that PPT is working. We have got another working group that is called Quality and Accreditation. Their job is to improve the quality of the blood bank. We are doing our bit, but we need the help of experts like you to do something. I mean, my only appeal to do something, or else we'll again come back after 6 months having another CME or webinar again, forget this topic again.
No,
So
Navjot, we'll take it up.
Yeah,
No, Navjot, we should take it up.
Yeah, I request, Ranjit to take up some sorts of activity time bound.
Sure.
So that, and we delegate each other, so what we can do by itself.
Sure.
Then only
No,
we
Navjot.
probably, that it is real requirement in this our member countries.
Sure. No, I
Except
think
few
Navjot,
countries, yeah.
We'll be more
Okay.
than happy to do it. I've been working with Thierry and Albert for almost 20 years, and they have been so helping, and Padma and Antesha, all of us will work together and see how best we can do it. So thank you very much, Navjot. And once again, I would like to thank all speaker and audience for making it possible. It was really, really very informative, very interactive session, and I'm sure we all were greatly benefited by the information which was provided. Thank you very much. I just will hand over to Kalyan for thanks. Kalyan, to you.
Thank you, sir. Thank you, Dr. Ranjit, for giving me the floor. And good afternoon, everyone, to one and all present here. Honorable Chief Guest and President, AATM International, Dr. Chaudhary, Dr. Sangeeta, our consensus chair of this AATM Plasma Protein Working Group, and all the participants. It is such an honor for me to get the opportunity to thank you all dignitaries. On behalf of Asian Association of Transfusion Medicine Plasma Protein Working Group, I, Dr. Roy, the secretary of this group, extend my warm welcome to the people in the gathering. I'd like to express my gratitude to all esteemed delegates of the webinar for their presence and contribution to make this webinar a great success. My hearty gratitude to our wonderful moderator, Dr. Ranjit Ajmani, for moderating the session in a majestic way. My special gratitude to Dr. Albert Ferrugia for his fabulous presentation, Evidence and Access, Current Issues in Plasma Products. Thank you for such an informative presentation. We're really grateful to you for your valuable time, sir. I extend my gratitude to Dr. Thierry Barnoff to take out time from his busy schedule to grace the event. Thank you for inspiring and encouraging with your mesmerizing presentation on access of safe plasma products on Asian perspective on this special day. A special thanks to Ms. Padma Sri Chigurapati for giving her time and being with us and discussing about the source plasma collection and challenges. Thank you, ma'am. I extend my sincere gratitude to Professor Anastasia and Dr. Bipin for giving their valuable time and made the panel discussion a memorable one. My wholehearted appreciation goes to all the audiences who has joined from the different countries around the globe. Your participation is the true inspiration to us to move on further. And last but not the least, I'd like to thank from the very bottom of my heart to Dr. Raikas Jindal, who has given us his endless technical support to us. Thank you everyone once again for making it a great success. Have a great day. Namaste.
Hi, Kalyan. Do not forget to invite everybody to your Congress in Bangladesh. You are the host country.
Yeah, definitely. I will love to invite you all because I think everybody know that there is AATM Congress is going to happen in Bangladesh, Cox's Bazar, in the upcoming December 1st, 2nd, and 3rd. So I welcome you all, and I'm sure you will enjoy. So just the registration is still open, and I think there will be spot registration are also available. So just be our guest and come to Bangladesh and enjoy. Have a great day.
Best
Thank you,
wishes,
Kalyan.
Kalyan.
Invite you all.
Thank you.
Okay,
Thank you all.
bye.
Bye.
Namaskar all.
Bye.
Bye-bye.
Invite
Very
you
much.
all, our
Thank
lovely
you.
Bangladesh.
Thank you.
Thank you.
Thank you.